Basketa. In this video, I'm going to say some few important topics on tissue chapter and fundamental unit of life chapter. See, in tissue chapter, what topic you should focus for your midterm is that in the beginning that you have a meristematic cells. See, in meristematic cells, you know that they don't have a vacuole. They don't have a vacuole. So what is the reason that they don't have vacuole? Because they do not have any role in storage of food. So what is the role? Their role is to divide, multiply. Okay, and you know that there are a different type of merisstems are there, like apical, lateral, intercalary. So you should know about their locations, right? You should know about their locations where they are located. Then you should know about simple permanent tissue like parenchyma, collenchyma, sclerenchyma. In that parent, colon and scleron, you should know their location and the functions of that, where this parent, colon and scleron are located. And everyone, like you know that sclerenchyma cells are dead cells. Correct or not? Sclerenchyma cells are what kind of cells? They are dead cells. Right? And parenchyma cells are present in soft part of the tissue, like in the root, stem, leaves, flower. And collenchyma mainly, collenchyma provide mainly a flexibility. Just we gave you that if you hang to a branch, the branch does not break it. It is due to the flexibility, due to the presence of collenchyma. Okay, so it is present in the leaf stack. Remember, leaf stalk. Leaf stalk. Which one? Collenchyma is present in leaf stalk. Okay, and sclerenchyma is dead cell. You know, it is due. It provides strength, right? It provides what? Strength, mechanical strength due to the presence of lignin. Then you learned about protective tissue in that epidermal tissue. Three modifications are there, like at the epidermis, cuticle is there, then stomata is there, then you have a cork cells, right? So you should know about, like stomata. What are the functions of stomata? You should know. So this is very important. Stomata helps in exchange of gases and also helps in transpiration, right? Transpiration. Opening and closing of stomata is controlled by who? Guard cells. So this you should know. You should know about complex permanent tissues like xylem and phloem. You should. What are the differences between xylem and phloem? So these are important things. Then if you come back to animal tissues, like you should know all type of animal tissue, epithelial tissues, squamous, columnar, cuboidal, ciliated and glandular, and compulsory they are going to ask like where they are located, location of this location, where they are located. Like ciliated epithelium is located where movement is required, like in respiratory tract, like in oviduct. Correct. So you should know about that, location. Stratified squamous epithelium, like epidermis of the skin, because it, where wear and tear is there, they are present stratified squamous epithelium, and even a glandular epithelium, just like where secretion of substance is required, there it is present. Right? So this location, especially the location, they will ask. And functions of epithelial tissue, four functions are there. Functions of epithelial tissue, if you read, you will, four functions are there. So those all four functions: protection, secretion, correct now, movement, removal of waste, so absorption. So those functions you can write. And another differences is like difference between bone and cartilage. This difference. Another difference is there, like a tendon and ligaments difference is there. So I think one question will come from tendon and ligament. And one more important topic is there, that is areolar connective tissue. What is the role of areolar connective tissue? That is very important. This areolar connective tissue is present just below the skin, remember. Okay, it is just present below the skin. What is the function? Like it repair the tissues, it fill the space, okay, it joins skin to muscles also, and it also kills the neuron diagram can come. Neuron diagram is very important, and they can ask about muscles like smooth muscles. See, these smooth muscles are involuntary. We are not, they are not under the control of us. So like in alimentary canal, the smooth muscles are there, remember. Okay. And then we have a stripped muscles, correct? Stripped muscles are striated muscles. They are voluntary. They are also called as skeletal muscles because they are attached to bone. Okay. And cardiac muscles are present in heart, that you know, very familiar one. Cardiac muscles are present in where? They are present in heart. So they can ask the difference between these muscles also. And rest all, like you know, neuron diagram. Peristaltic movement, remember, peristaltic movement is due to the smooth muscles, remember. Peristaltic movement is due to which muscles? Smooth muscles. So I don't think so other than that anything is possible in that. And in fundamental unit of life, they can ask definitely about you know, what is plasmolysis. They can ask plasmolysis. Main topic, plasmolysis is there. Plasmolysis is nothing but when a cell is placed in a hypotonic solution, water enters the cell. Okay, so when water enter, it will get what you can swells and right, and comes in contact. Yeah, see, actually what plasmolysis matlab, like it is placed in this solution, hypertonic solution. Yeah, it is placed in a plant cell is placed in a hypertonic solution, then it will get shrink. Okay, that shrinkage of cell when it is placed in a hypertonic solution, it is called what? Plasmolysis. And then if you place in a water hypotonic solution, it will get swell up and again it will become a normal cell. Okay, so that is in that prokaryote and eukaryote differences they can ask. What are the prokaryotic cell and eukaryotic cell differences? They can ask, you know that. So that is a topic of prokaryotic and eukaryotic differences. And one question is compulsory on plasmolysis, or you can say what is hypertonic solution, hypotonic solution, and isotonic. These are the three type of solution. Hypertonic solution matlab you know na. Hypertonic solution matlab what? What is the meaning of hypertonic solution? Anyone knows here? See, hypertonic solution matlab what? A solution where water concentration is less and salt concentration is more. Correct? If for example one solution is there, if here salt concentration is more and water concentration is less, then from the cell water comes out. If from the cell water comes out, this process is called plasmolysis, shrinkage of cells. Okay, and it is happened when it is placed in which? Hypertonic solution. Hyper matlab water is more and salt is less. Okay, so there swelling up takes place in the hypotonic solution. Iso matlab salt and water concentration is same, no movement takes place. Okay, you should know about nucleus diagram there. Okay, nucleus diagram. And you know why the lysosome is called suicide bags of a cell? Why lysosomes are called suicide bags of a cell? Why they are called? Because they can, you know, when anything abnormal activities takes place in a cell, then this this lysosome burst, and you know, I mean, they burst and releases enzyme and digest its own cell. And you should know about difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. So this is very important. What is the role of endoplasmic reticulum? Protein synthesis is there, right? Even you should know about the Golgi apparatus. Golgi apparatus function is what? You know, they mainly plays the important role in transport. Okay, packing and transporting, you can say. So those things. Ribosomes already you must be knowing. Protein factories, mitochondria is very important. You know, powerhouse of cell. And you know that who is having their own DNA, correct? Who is having own genetic material? Mitochondria is having their own genetic material. Even chloroplast is having their own genetic material. So chloroplast is present only in the plant cell, remember. Chloroplast is present only in plant cell, not in the animal cell. So this thing only, and that's it. This much you prepare it, I think you will be. And read our notes. If you don't have, ask to someone to send a pic of those. You know everything is covered in that. I hope you will get a good marks.